Good afternoon, everyone. My name's Professor Sharon Lewin, and I'm the Director of the Peter Doherty Institute for Infection and Immunity, and delighted to welcome you to Monday lunch live stream at Peter at VCCC. I'd like to start by acknowledging the traditional custodians of the lands on which we meet today. I'd also like to pay my respect to elders past, present and emerging, and welcome any Indigenous Australians with us today. Now, you might be thinking to yourself, why is Sharon Lewin hosting a VCCC seminar? Well, I'm delighted to host this seminar because we'll be talking about and hearing from Joe Trapani about the Centre for Cancer Immunotherapy, and I'll talk more about that later. First of all, just to remind you that VCCC has some very interesting speakers coming up, so please register your interest via their website. They also have a learning page if you'd like to view previous topics. Now, just to set the rules for today's seminar, which I'm sure all of you are very familiar with, as we've all spent way too much time on Zoom and on too many Zoom webinars. But if you have any questions for today's presenters, you can do this via the Zoom Q&A function. Please submit your questions through the presentation and we'll aim to answer your questions at the end of the session, and I'll keep very close track of what's coming through and hopefully allow everyone to have their questions and finish in time to end this webinar at 2 p.m. So it's my very great pleasure to introduce our two speakers for today, Professor Joe Trapani and Professor Paul Neeson. So Joe, as many of you will all know, Joe, of course, is head of the Cancer Immunology Program at Peter MacCallum Cancer Centre, was previously the Executive Director of Cancer Research and the inaugural head of the Sir Peter MacCallum Department of Oncology at the University of Melbourne. And in 2018 was appointed Director of this new initiative, the Centre for Cancer Immunotherapy for Peter Mac and the VCCC partners. And Joe has done a fantastic job at reaching out not only to VCCC partners but beyond, including the Doherty Institute. As many of you know, we work extensively in immunology, not just immunology of infectious diseases, but autoimmunity and, of course, high interest in immunology and immunotherapy for cancer. So we're very excited to do even more with the Centre for Cancer Immunotherapy, and hopefully may even have some Doherty Institute staff based over at the VCCC in coming months, which both Joe and I are very excited about. Joe's research interests include the immunopathology of viral and autoimmune diseases, apoptosis, and cancer immunotherapy. Most recently, he's become a pseudovirologist, I should add, because we've been working with him on a really interesting project on SARS-CoV-2 as well. He's authored more than 310 research papers, reviews, and book chapters, and participates in many other peer-reviewed bodies for state and federal governments and in academia and industry. So it's my very great pleasure to hand over to Professor Trapani, and Joe will also then formally introduce Paul Neeson. Over to you, Joe.
Well, thanks very much, Sharon, and I can't see anyone, but I want to welcome everyone, and I hope you can hear me. I assume that if there's some problem, Cynthia, my friend Cynthia at VCCC will let me know. So thank you for joining us. And really today I'd like to talk about our research program in CAR T cells, or chimeric antigen receptor T cells, and particularly the emphasis is on how we're trying to extend the success that's been achieved thus far in certain cancers, particularly B-cell cancers, so childhood acute lymphocytic leukemia, for example, and some B-cell lymphomas, to the much harder problem of common solid tumours where the success rate really remains disappointingly low. And just to remember that it's now been probably 7 or 8 years since the first extraordinary trials in childhood ALL were reported. We think there are a number of reasons for this difference. Probably one of the key differences between ALL and the solid tumours is the suppressive tumour microenvironment that we have to overcome in solid cancers. And another is probably, I believe anyway, the choice of antigen that we choose to target. And today I'll concentrate for a number of reasons on an antigen that we've worked with for many years, and indeed taken from lab research all the way into our own clinical trials, and that's an antigen called Lewis-Y. So given the diverse audience today, I thought I'd use just a couple of minutes to make sure you're all aware of the VCCC Centre for Cancer Immunotherapy. This is really quite important, and we're looking to make connections, and particularly with clinicians, and to set up new collaborations all the time. This is a major collaborative venture of the VCCC partners, and it only got going just under 2 years ago in November 2018. After that, I'll introduce CAR T cells and talk a bit about the history, not just for history's sake, but because this too is steeped in some fantastic collaborations between a number of what are now VCCC partners over the years. So to some extent, my talk today is really sort of retrospective, but there's lots of fantastic new stuff happening in CAR T cells, and my colleague, Professor Paul Neeson, will outline some of the current projects in this area. Next, please. So what is the Centre for Cancer Immunotherapy? Well, it's partly physical space because the government, the state government, was kind enough to build us some labs up on level 13. But really, it's very important that most of the Cancer Immunotherapy Centre is actually virtual and really extends across the whole alliance, the whole partnerships, and indeed beyond. In preparing for this talk, I was looking for something about the vision. And in the end, I remembered that I should go to some slides that Sean Grimmond and I prepared when we made our pitch to the VCCC board back in 2017. And ultimately, I found the vision in one of those slides, and you can see it down there towards the bottom in blue. So this is very, very, very important. What we really would like to do is that within a couple of years, every patient admitted to a VCCC affiliated health service, and I'll remind you that of the 10 partners, 7 are Victorian public hospitals. So that's really important. Anyway, every patient, where appropriate, would be assessed for their suitability to participate in an immunotherapy trial, or if there's something that's already known to be effective or to help, then the optimal combination of immune and other therapies would be offered to that patient. Next, please. So just very briefly on sort of resourcing and the model. So as often happens, government funds you, you get a great capital budget, and we're very grateful for the labs, but usually you don't get a recurrent budget for research activity. And here I'm very, very grateful to the VCCC board and to the Cancer Research Advisory Committee for approving some funding from the Strategic Research Plan for the VCCC. And with that, we've been able to fund a full-time clinical fellow, a fantastic fellow, Dr. Minyu Wang, and a bit of RA support, which we've had going for the past year and a half or so. As will become relevant in a moment, both the university and Peter Mac have also committed some funding for recruitment. My role is very much to foster collaboration, to talk to people, to really make sure we're working mainly on tumour stream-based research, and we're really trying to concentrate initially, in particular, on areas of important unmet clinical need, and as a priority. And my role is also to leverage funding, to bring in-kind resources where we can, and so forth. Part of the model does include co-locating small research groups from any of the interested VCCC partners who may wish to join us up on level 13. This wasn't that easy operationally, but we've really got that working pretty well now. And so why would you want to put people up there? Well, it could provide you with access to patient material and data, certainly to expertise and lots of assays and so forth in cancer immunology. And Paul runs some very sophisticated research platforms, which would also be important. And then just for completeness, I report through a 4-person senior advisory group, Grant MacArthur, Andrew Scott, Sharon and Ricky Johnston, through to the VCCC board via the Cancer Research Advisory Committee. Next, please. Okay, just a touch on progress. I think we've made a solid start. There are 4 collaborative projects that are well and truly running, and I'm very delighted with one in particular in head and neck cancer with the Royal Melbourne and David Wiesenfeld. We're looking at a very interesting project with the WEHI on advanced melanoma, a lot going on in prostate cancer. And as you'll hear a little later, we're doing quite a bit in kids cancer, a terrific collaboration with the Murdoch and the Royal Children's in AML and high-risk neuroblastoma. We've relocated small groups from Olivia Newton-John, the WEHI, the Murdoch, and as Sharon mentioned, we're really delighted that it's very likely that a small group will join us from Peter Doherty Institute and University in the next few months. We've started to bring in some grants, and we're looking forward very much. We're participating very actively in the next research strategic plan for the VCCC, and we're talking with Grant and JH Desai and others about increasing and improving yet further the integration with clinical trials research in particular. Now, an important achievement has been with the support of Peter Mac and the university to make a very exciting new appointment for a role called the Head of Clinical and Translational Immunotherapy at the Centre. Next slide, please. And so it's a great pleasure. The embargo was finally broken this morning. It's a great pleasure to introduce, in a virtual way, the inaugural appointee, who's Professor Riccardo Dolcetti. Riccardo is currently Research Chair of Cancer Medicine at the Diamantina Institute in Brisbane, but will be joining us in the next month, if we can get him down here with the pandemic, down here in Melbourne in the VCCC building. Riccardo is really a very, very highly qualified breast cancer oncologist and lab-based researcher and translational researcher. He was previously director of a very significant cancer immunotherapy institute in northern Italy. He's had a professorial appointment in the United States. He's a great leader and mentor, and that's reflected in the fact that he was president of the Italian Society of Cancer Research some years ago. So, Riccardo, we're delighted that we've been able to announce this appointment today, and we look forward to seeing you in person down in Melbourne very, very soon. And we may be able to say hello to Riccardo, perhaps if we can hook up at the end of this talk. Next, please. Okay. So now for a bit of science from me, and really I'd like to go back, and many of you may not know that we've actually worked on CAR T cells since the mid-1990s, one of the first groups in the world to do that. So why on earth did we do that? Well, it was a bit countercultural at the time. My group was actually out at the Austin Research Institute in those days, which was co-located very close to the Ludwig Institute on the Austin campus. And we really made the strategic decision to go against sort of the conventional wisdom. And no one was really trying adoptive immunotherapy very much in Australia. There were many, many attempts at active immunisation for cancer, and that had been literally tried for nearly a century since the time of William Coley. And we decided that we'd try adoptive immunotherapy. Trouble was, adoptive immunotherapy at that time wasn't working very well. I'll show you just an example or two in a moment. It was called TIL therapy. So TIL, still a very, very relevant term, tumour infiltrating lymphocytes. And this really had a lot of drawbacks. Our strategy, in fact, was to address these drawbacks using gene therapy and to particularly to express antibodies within the T cells to try and redirect or retarget them to cancer. Next, please. So just to remind you very briefly, in fact, TILs have been used as therapeutics since the 1980s, particularly through the fantastic work of Steve Rosenberg and his group at the NCI. In TIL therapy, the lymphocytes are actually taken out from a fresh surgical specimen of the tumour. So there's a requirement that the patient has undergone recent surgery. And that does one good thing. It does remove them from the immunosuppressive environment of their tumour. They're cultured ex vivo in clean rooms, expanded, activated, and with T-cell growth factors, particularly interleukin-2. And then they're given back to the patient as a vaccine, and that might take a week, or sometimes it might take quite a lot longer. Next, please. And double click. One more click, please. So, in fact, there have been probably hundreds of TIL trials, but even today, TIL vaccines are not really widely adopted, although they're modified and they're coming back in other forms. But nonetheless, the reason they haven't been widely adopted is that their success rate with this type of technology was actually quite low, only about 5%. But quite remarkably, in some of these cases, we saw an amazing amount of tumour destroyed in a very short period of time. And just a couple of examples up on the screen. There's a radiological one on the left, but if we just look at the picture of the patient here who has disseminated widespread melanoma with 2 huge subcutaneous metastases, the larger one of these surely must have contained many grams of tumour. And if you look at the inscription just above the picture, you can see that just 12 days after receiving a single dose of TIL therapy, both of these lesions are completely gone. Now, this happened too many times to be coincidental or accidental, and it was important proof of concept to us that perhaps if we got the conditions right, the approach might work. Next, please. I mentioned TILs had lots of limitations. Not all patients' tumours even have TILs. It might take weeks or months to expand out these rare clones. A big problem was IL-2 toxicity because you had to give typically IL-2 to sustain the CD8 T cells in vivo, and IL-2 is toxic to many patients. And then there's the issue of relapse and loss of class 1 and so forth. Next slide, please. So our sort of simplistic proposition was that a CAR T cell effectively would be, they weren't called CAR T cells in those days, of course, but the vaccine would be a patient's T cells redirected to their cancer with antibodies. That was the basic proposition. Now, you've got to remember that back in the 1990s, if you're old enough to remember back that far, monoclonal antibodies were very exciting. They were brand new therapies. They were being called magic bullets. The idea being that they would carry really toxic drugs or isotopes in high concentrations to the cancer, but spare normal tissues because they were so specific in what they could target. Now, I said we were at the Austin. My boss, Ian McKenzie, shown there on the left, was making immunoconjugates for a good 10 years by then. And a few years later, so was Professor Andrew Scott, who remains a collaborator to today. And in fact, one of the targets shown at the bottom there that Andrew and his colleagues at the Ludwig and originally back at Sloan Kettering actually was aiming at was an antigen called Lewis Y, and that's actually relevant for later in the talk. Next slide, please. So I mentioned Andrew's still very much part of the collaboration, and even these days, Andrew is still used as an absolute champion in immunoconjugates, particularly when it comes to diagnostics and so forth. Andrew reminds you, in fact, that a CAR T cell is just a special immunoconjugate, and in fact, he's right. So we can compare and contrast CAR T cells and conventional immunoconjugates in many ways, as shown in this table. But the crucial difference is the payload that the antibody carries. And if that happens to be a really well-activated killer T cell, then the vaccine does some very special things. For example, it'll continue to renew itself in the patient. And it'll engage with the rest of the patient's immune system to do things like what we call epitope spreading, spreading out to other things that the cancer expresses. And then you get immune activation, recruitment of the patient's own immune system, and things like recall and memory responses, which are critical for long-term success. Next, please. I really wanted to mention the contribution of a couple of key people along the way. The first is Mike Kershaw, who's now Professor Mike Kershaw, group leader in our program. Mike really was, without Mike, I don't know that we ever really would have got this program off the ground. He actually came to my lab as a mature age student way back in 1993. I should note that that's not a picture of Mike in 1993. He wasn't quite that mature. That's a recent picture of Mike. But Mike's PhD thesis, called Redirection of Cytotoxic Lymphocytes by Genetic Manipulation, was one of the first in the world on CAR T cells. As Mike wrote in the opinion piece there, the redevelopment of re-engineered monoclonal antibodies with tailored properties and their characterization of newly defined endogenous tumour antigens has brought us to the brink of using these cells for therapy? Well, it took us another 10 years, but we did get there. And you'll notice in the box at the bottom that one of the possible structures that Mike alluded to was in fact the modern chimeric antigen receptor with a couple of tiny modifications. Next, please. So the second person I'd like to mention is Phil Doherty, who joined us from St. V's, or St. V's Institute, in 1995. Now, I'm not sure how widely it's known. In fact, the first paper, genuine paper on CAR T cells in cancer came from an Israeli group at the Weizmann Institute, led by Zelik Eshar. And we collaborated and spoke very often to Zelik, and he was incredibly helpful. But within a couple of years, Mike and Phil and others were well and truly in the race. Next, please. Next slide, please. Thank you. So in total, we've published many papers, but I wanted to highlight two key early discoveries. The first was in two papers by PhD student Nicole Haynes. Nicole is still with us and going great guns. And she showed that in order to get maximum potency, you needed to engineer into the CAR signaling domains a co-stimulatory signal, as well as one that mimicked T-cell receptor signaling. Next, please. So here's a picture of Nicole. And in this experiment, which is taken from one of her papers at that time, we see in the green traces that mice that are bearing HER2 positive or ERB2 positive tumours survived best after a single dose of CAR T cells if the signaling domain had both TCR and co-stimulatory domains, that's shown in green, as compared to just having one, as shown in orange. And indeed, if you delayed the therapy, where you can see the black arrow at the bottom, of course, the greater the delay in the therapy, the less the therapeutic effect. But in every case, mice that received the two-tailed, what we call the two-tailed receptor, did best. Next, please. Okay. The second one, which is another very, very significant advance, was made by another PhD student, Maria Muller, shown here with what looks a bit like a halo. Maria was another PhD student, she made the critical observation that to get a therapeutic effect, and this was really rather counterintuitive, at least a proportion of the T cells, of the CAR T cells, indeed, in this model, 50% was the best ratio. They actually had to be non-cytotoxic CD4 cells. And in fact, their indispensable role was to make cytokines, particularly interleukin-2, to sustain the cytotoxic CD8s. Quite remarkably, if you look at where those vertical parts of the curve are over in the bottom graph, if the vaccine was 100% fully activated CD8 killer T cells, 100% killers, you got no therapeutic response whatever. You must have cognate CD4 help. It has to be directed. When I say cognate, non-specific help won't do. If you put in a CAR T cell with specificity for a different antigen, again, this will not work. Next, please. So this was a very original finding at the time, and in fact, we patented the finding, and this was a very important decision to make because subsequently our patent was licensed by Juno Therapeutics, and they funded a large clinical trial led by Ben Solomon, and that work has been very, very instrumental in other collaborations and papers and grants. Next, please. So the final bit of the puzzle to get CAR T cells into the clinic was to have access to clean rooms for vaccine manufacture. Now, this was actually, of course, a picture of the amazing rooms we have in our current facility. But one of the reasons our program moved to East Melbourne back in 2000 was to have access to the forerunner of this facility, which of course was far humbler. But really we have to remember the vision and thank the people who were key to this decision. And they included Professor John Salzberg, Miles Prince, the senior management of Peter Mac at the time. John Morris was the CEO. That was actually, the facility was opened by Prime Minister John Howard. And I'd also like to acknowledge the wonderful work of our Peter Mac Foundation. Next, please. Okay, then just to remind you, here's a CAR T cell, diagram of a CAR T cell. We use retrovirus or lentivirus to express the chimeric receptor, and we repurpose the killer T cells, which are harvested from a patient's peripheral blood. to recognize cancer cells. Now, like the chimera from the real chimera, the original chimera from Greek mythology 2,500 years ago, the chimera doesn't actually exist in nature. It's actually synthetic biology, and it's linked. So there are several different domains, artificially linked. So externally, we have a single chain antibody, a transmembrane domain, and then the two signaling domains that I mentioned before, TCR signal and a co-stimulatory signal, CD28, 41BB or something else. Next, please. And the difference is that if you get really good connection, no, back one, please. If you get really good connection with the cancer cell, then the things that pop up in blue on the left are very important. You'll get killing of the cancer cells, you'll get proliferation and stimulation of the CAR T cells within the patient. They'll recruit the immune system, the endogenous immune system as well. You'll get inflammation. And then if you're going really well, as long as there is antigen around, in principle, the CAR T cells should persist and you should be able to recall them as well. Next, please. So as I mentioned earlier, you might click that, I think, 3 more times, please, Cynthia. Good memory. Now, as I mentioned, some years ago, the US FDA approved initially the Novartis CD19 CAR T cell product for the treatment of refractory pediatric acute lymphocytic leukemia. And I always love to show this slide because every year I go back to the Emily Whitehead Foundation website, and she looks a year older but just as healthy. So here's Emily back in 2010 when she was diagnosed with ALL at the age of 5. She fell into the 10 or 20% of kids who do badly. Thankfully, most kids do well with standard therapy. But she really was doing extremely badly in 2012. She was enrolled in the CAR T cell trial. She got better and she's healthy 8 years later. Next slide, please. Now, unfortunately, no solid tumours express CD19. And if we're going to make headway in lung, colorectal, pancreatic, or other cancers, we need a good target. So this was actually an old slide of mine. I think I made it about 2004. And it's sort of hypothetical. If you could design the perfect target, what would the ideal target look like? What would be its properties? Well, they're here. The target would be abundant on tumour cells. It would not be shed so that you don't block the CAR T cells when they're circulating. The target would not be on normal tissues, would not be easily lost or subject to mutation, and it would be great if it was expressed on many different types of tumours or classes of tumours. Next, please. So in fact, one of the antigens that really, I think, meets most of these criteria is one I mentioned a little earlier, Lewis-Y. It's a really unusual antigen. It's not a protein, it's a glycolipid, and it decorates many, many proteins, carbohydrates, and other lipids. And for that reason, it's very, very abundant. There are millions of copies of this epitope on about half of many common adenocarcinomas. And it doesn't matter whether it's upper lower GI, breast, prostate, lung, pancreas, etc. And important for our first trial, about 10% of patients with AML, acute myeloid leukemia, have a Lewis-Y positive, but at very low levels, unfortunately. It's a neo-antigen. It's only expressed in the embryo and fetus, and it's switched off after birth. And in fact, because Andrew Scott and colleagues in many parts of the world had used the parental clone to carry drugs and radioactive payloads to tumours, we knew that that was probably right. The tissue distribution and the toxicity we anticipated would be very manageable. And you can see at the bottom, intense immunohistochemical staining in a primary lung cancer. Next, please. So we were very grateful when the Leukemia and Lymphoma Society funded our first human trial, which happened to be in AML, as I mentioned. We enrolled 7 patients, eventually 5 got the vaccine. And of course, as in any phase 1 trial, the first aim and main aim was around safety, but we were also interested in other surrogate markers. Would the T cells co-localize with tumour? Would they persist? And so on. Next slide, please. So very, very briefly, clinical summary, all 5 patients had advanced disease. that all failed other forms of therapy. The therapy was extremely well tolerated. And just in that red box, you can see we had one patient, interestingly, developed a fever and a rash that I'll come back to. Two patients had transient reductions in tumour mass, but they were probably of no real significance. Next slide, and we'll go rapidly now, Cynthia. A lot was achieved. By PCR, we showed long-term persistence of the CAR T cells in peripheral blood and bone marrow, one patient for up to 10 months. Next. In several, we saw considerable spikes of circulating cytokines, including interferon gamma, IL-2, IL-4, and down on the bottom right, the spike of TNF in the bone marrow of one of the patients. Next. And this is actually quite important. So I mentioned that one of the patients developed a rash. So 6 days after receiving her vaccine, this patient, patient number 3, developed a tender nodular rash. And when biopsied, what it showed, both by PCR on the bottom left and then by immunohistochemistry, was co-localization of leukemic cells that had been infiltrating the skin, co-localized with CD4 and CD8 CAR T cells. So in fact, leukemic cells had not been suspected in the skin of these patients. This is a condition called leukemia cutis. But in fact, they were revealed by the inflammation caused by our vaccine. And that was actually, I think, a very, very interesting thing to find. Now, nearly ready to hand over to Paul. Next slide, please. There are way too many preclinical approaches being explored at present to discuss them at length, but some are just listed here, and Paul will pick up on some in some detail after me. But basically, we're trying to improve T cell quality in various ways, tackle the suppressive tumour microenvironment, and importantly, engage with endogenous immunity so that we can get things like epitope spreading and recruitment of the patient's immune system itself. Now, one strategy we're very keen to advance, in fact, is to combine CAR T cells with conventional therapies that patients are going to be getting and that are potentially inherently immunogenic. And one that's obvious is radiation therapy. And particularly focused radiation, for example, as is given by SABR, or stereotactic radiation. Now, great news, and it's embargoed at present, but I think very soon you'll hear some good news about a big collaborative grant that I think will be supporting us to be able to take these studies a bit further. Next, the final thing I wanted to talk about, and if we could click that twice more, please, is that if you wish to follow up some other recent work, which is very recently published, that we won't be covering today, please look up Paul Beavis's paper, very recently published in Nature Immunology, along with two fantastic PhD students, Shirley Marianne and Jun Yun Lai. Paul's CAR T cells express FLT3 ligand, and they recruit dendritic cells to the tumour. And next, finally, a terrific paper also from Claire Slaney shown there. And Claire is using a parallel and quite different approach and collaborating actually with Riccardo Dolcetti using his nanoparticle technology for vaccination and recruiting the immune system, again targeting dendritic cells. And then I think the next is my final slide, and this is really just to introduce Paul Neeson, and I'm sure Paul will also introduce Deborah and Paul Eckert. Thanks, and over to Paul.
Thanks, Joe. This is the first of two projects I'm presenting today that Deborah Mayron has established in the lab. And this first one is targeting the Lewis Y antigen with CAR T cells in pediatric AML. So the aims of the project were to investigate Lewis Y expression on pediatric AML blasts by examining primary patient AML bone marrow samples, and then to characterize functional responses of the Lewis Y CAR T cells to these pediatric primary AML blasts, and then finally to investigate in the pediatric normal tissue the distribution of the antigen Lewis Y. So Deborah showed unequivocally that AML blasts represented here in FACS plots in this gate express the Lewis Y antigen on the y-axis here, but the normal lymphocytes do not. And furthermore, when she examined 10 samples, she was able to show that all 10 samples expressed Lewis Y antigen on the gated AML blasts. Interestingly, there was a spectrum of expression of Lewis Y antigen on the AML blasts everywhere from low levels up to high levels based on mean fluorescence intensity. So then we examined the functional responses of the AML blasts, of the Lewis Y CAR T cells to these AML blasts. So we're looking at the first patient C975, as the target for the CAR T cells. We can see that this is a percentage of killing of the target cells by the Lewis Y CAR T cells, that indeed the CAR T cells can kill the target 975 expressed at high levels of Lewis Y. Interestingly, also another target, C035, a different patient who had intermediate levels of Lewis Y, those AML blasts were also killed by the CAR T cells. The other interesting aspect that Deborah's exploring is the relationship between this specific MLL rearrangement in ALL, AML, and expression of high levels of Lewis Y. And she's shown a series of cell lines derived from patients with the MLL rearranged ALL, all expressing high levels of Lewis Y. Then returning to screening normal patients. So she screened 10 healthy donor bone marrow samples and demonstrated there was no Lewis Y antigen on myeloid blasts. This is the myeloid blast here showing no evidence of Lewis Y. So in summary, at this point with the development of this project, we know that pediatric AML does indeed express high levels of Lewis Y in approximately half the patients, and it appears to be at much higher levels than it is in adult AML. We know that Lewis Y CAR T cells kill Lewis Y positive pediatric AML blasts, even with intermediate level antigen expression. So we have this open question now to explore whether this increased Lewis Y expression is associated with MLL rearrangement. So our ongoing studies include the in vitro characterization of the Lewis Y CAR T cell responses to pediatric AML to confirm, importantly, that the normal tissue in kids does not express Lewis Y and can therefore not be a target for Lewis Y CAR T cells. The second project that we're exploring currently is overcoming obstacles to CAR T cell efficacy. And Joe mentioned before two of the exciting projects that are ongoing and published already in the program. Key things for CAR T cells to overcome include tumor heterogeneity. In other words, that there are multiple antigens that need to be targeted within the tumor microenvironment. Furthermore, CAR T cell trafficking to solid tumors, particularly into peripheral tissue, that once the CAR T cells get there, that there is many mechanisms of immune suppression which reduce the efficacy of CAR T cells at that site. And importantly, persistence of the CAR T cells long-term in the patients after they're actually infused. We know already that increased T cell memory compartment in CD19 CAR T cell product is associated with CAR T cell persistence and long-term remission in patients with acute lymphoblastic leukemia. But we don't know whether this is true or not in solid tumors. At this point, we're just completing now our second trial with Lewis Y CAR T cells, and we've treated 20 patients using the second generation Lewis Y. And using samples from this particular clinical trial, we've examined the peripheral blood after infusion of the CAR T cells to see if the CAR T cells persist in patients with solid tumors. As I mentioned before, at this point, that when the CAR T cells are reinfused into the patient receives a lymphodepletion regimen, which enables the CAR T cells, once they're reinfused, to undergo homeostatic proliferation and expansion. And indeed, that's what we observe. So this is transgene copy numbers per microgram of DNA, or the number of CAR T cells per mil of peripheral blood. And you can see for each patient, there's a curve where there's a rapid expansion of the CAR T cells, a plateau, and then a slow, steady decrease of the CAR T cells over time. So we are developing a project where we're investigating an optimized CAR T cell protocol to address CAR T cell persistence in vivo. So the upper half of this figure depicts the conventional CAR T protocol, where we take apheresis product and we activate the T cells and retrovirally transduce them and then expand them in high dose IL-2. And for the remainder of this talk, we've broken up T cell differentiation based on color. So the earlier the T cell type, the more blue it is, the more differentiated T cells are more purple. So this conventional CAR T cell largely produces T effector memory cells and very small memory component. In contrast, Deborah and Joe developed a very brief production protocol where they initially isolated naive T cells from the PBMCs. They activated and retrovirally transduced them and then expanded them for just 3 days in a memory IL-7, IL-15 cytokines to generate more of these early cells. We call this the early CAR T protocol. And then we characterize the sort of cells that we produced out of this. So we find that they are increased for a combination of two markers, 62L and CD45RA, which are classically expressed on early cells. So we see after a number of transductions that the early CAR T cells have significantly increased numbers of stem cell memory-like T cells, and in contrast, the conventional CAR T cells have a significantly increased number of effector memory T cells. To characterize these early CAR T cells further, in order to take it further towards the clinic, we then used mass cytometry and used a large panel of markers to find these T cells. So we used a panel that included lineage markers, differentiation, activation, proliferation, expression of the CAR, immune checkpoint molecules, effector function, and transcription factors. And we showed unequivocally when you plotted each cell with the 40 parameters on a two-dimensional UMAP that the conventional CAR T and the early CAR T were completely different. And furthermore, if you superimpose T cell differentiation on top of that, we can see on the left-hand side here are the early CAR T, that they are enriched for stem cell memory and central memory in both the CD4 and CD8 subsets. In contrast, the conventional CAR T are enriched for a much more differentiated pool of TEMRAs and T effector memory, and that's represented on the right-hand side as well. Then if you displayed some key markers of T cell differentiation, including 62L, TCF7, CD27, we can see that the early CAR T cells are enriched for these markers and they're also proliferating. In contrast, the conventional CAR T cells have higher levels of perforin, so more of an effector phenotype. Importantly, we're looking at also immune checkpoints because we don't want to put exhausted T cells into patients. We want to generate CAR T cells which have more functional capacity. So we can see the distribution of the early CAR T cells are on the left here, the conventional CAR T cells on the right. So we can see that the early CAR T cells have very little LAG3, PD-1 and TIGIT, although they do express some T3. In contrast, the conventional CAR T have high levels of LAG3, PD-1, TIM3 and TIGIT. To test the function of these this new production of early CAR Ts, we co-cultured the cells with tumor lines that were Lewis-White positive or negative and looked at degranulation or cytotoxicity after 4 hours. And we showed that the early CAR T cells were excellent at killing Lewis-White positive targets, and as are the conventional CAR T. And furthermore, that the early CAR T degranulate significantly higher in response to Lewis-White targets versus to Lewis-White negative targets. We also showed importantly that the early CAR T cells are polyfunctional and secrete significantly higher levels of cytokines than conventional CAR T cells. To do this, we had the same co-culture system, but collected the culture supernatant and measured cytokines after an overnight co-culture. And we showed that indeed the early CAR T cells secreted higher levels of interferon gamma, TNF alpha, and IL-2. In a 7-day proliferation assay where we had the early CAR T or conventional CAR T in just homeostatic cytokine IL-2, or where we stimulated the CAR T cells via the chimeric antigen receptor, we can show that the early CAR T cells were much better at proliferating. They had significantly higher proliferation than the conventional CAR T when the cells were stimulated via the CAR. So in summary, conventional CAR Ts, where we transduce PBMCs, generate effector memory and T central memory cells at day 14. When we transduce naive T cells and generate the early CAR T cells at day 6, these cells are enriched for stem cell memory and central memory cells. They have antigen-specific cytotoxicity, and they have increased antigen-specific degranulation following long-term co-culture with Lewis-Y positive targets. And also, they show increased antigen-specific proliferation. In addition, other studies in the lab have shown, there's no time to show you this today, but that we can actually chronically stimulate these cells via the CAR for a month. And these early CAR T cells have much better, significantly increased functional responses and retain their memory pool after 1 month of stimulation. And finally, the CD4 T cells are required in the naive pool to generate effective CD8 positive CAR T cells. So then we took our findings into a mouse model where we have the Lewis-Y positive line of CAR3 inoculated into NSG mice. 2 days before CAR T therapy, we irradiate the mice, and then we inoculate the mice 3 times with CAR T cells. We monitor tumor growth kinetics subsequently, and we look at we collect peripheral blood at day 15 for looking for trafficking of CAR T cells and persistence. The CAR T cell type that was put into the mice included the conventional CAR T at a high dose, early CAR T at either a high or low dose, and then control CAR Ts. The data show that the early CAR T cells maintain better control over Lewis-Y positive tumor growth than conventional CAR T cells. And this was particularly true for the high dose early CAR T, and they were much more effective than the conventional CAR T cells, which were more effective than the needed controls. In addition, the early CAR T at high dose had those mice that received that therapy had the best survival. So peripheral blood correlate of persistence of these CAR T cells showed that the high dose of early CAR T cells was correlated with increased frequency of circulating CAR T cells. And furthermore, when the tumours were measured at that time point, an increased frequency of early CAR T cells circulating was associated with shrinkage of the tumour. Because CAR T cells upregulate PD-1 once they sense their antigen, we wanted to explore whether we could combine immune checkpoint blockade with CAR T therapy. For that to occur, we needed to show that PD-L1 could be expressed by the target line of CAR3. Whilst of CAR3 don't express PD-L1 constitutively, when we took of CAR3 cells and co-cultured them with CAR T cells and then collected the supernatant and added this conditioned medium to of CAR3 for 24 hours, we had large upregulation of PD-L1. In fact, this was even more so than just interferon gamma alone. So then we had a rationale for testing the combination of early CAR T cells with the anti-PD-1 in tumor-bearing mice. So this was once again the NSG of CAR mice, and at the time of injection of the CAR T cells, we added anti-PD-1 or early CAR T cells alone, or the control T cells plus anti-PD-1. We monitored tumor growth kinetics and then also looked at peripheral blood at day 15 for trafficking of persisting CAR T cells. And we showed that the combination of early Lewis-Y CAR T cells plus anti-PD-1 eradicated established of CAR3 tumours. So this is complete eradication of the tumours in the red line when anti-PD-1 plus Lewis-Y CAR T, and then the early CAR T cells alone here in the blue, and then our controls, untreated or controlled CAR T cells. At the same time, we looked at day 15 and showed that there are persisting early CAR T cells in the peripheral blood, and that these were increased in number compared to the anti-PD-1 combination with early CAR T. Interestingly, the early CAR T plus anti-PD-1 mice, treated mice, they retain their stem cell memory-like pool, whereas the early CAR T cells started to convert to central memory cells. And this data was reproducible across multiple mice. At this point, we're looking at, in another series of experiments, the CAR T cells that have penetrated the tumour and are comparing the differentiation and functional responses of those cells to those that are moving in the periphery. We've taken this concept now into patient setting, and we've actually produced early CAR T cells from a cancer patient's PBMCs. This data here shows the phenotype that we can generate memory cells using the early CAR T protocol, and they are completely different to the effector memory type that you see in conventional CAR T protocol. And furthermore, these same early CAR T cells are much better at proliferating than the conventional CAR T cells when they're produced from a cancer patient's T cells. Our second strategy is actually a new CAR construct using truncated human CD34, which enables selection of CAR-positive cells and anti-tumour responses. So here we can actually enrich for CAR-positive T cells at a lower cell dose. So the clinical problem with efficacy of CAR T cell therapy for solid tumours is that there's a lack of persistence, and this is due to the low memory pool in conventional CAR T product. The early CAR T strategy generates increased stem cell memory cells and central memory cells, better de novo function, better function after chronic stimulation. We know the CD4 T cells are required. We have better anti-tumour efficacy. We can regress tumours when combined with anti-PD-1, and we can produce early CAR T cells from solid cancer patient T cells. So our new strategy now is to generate clinical grade early CAR T cells is to actually use naive T cells from the bulk PBMCs, activate, transduce, and then expand in the memory cytokines, and then reinfuse these early CAR T cells following lymphodepletion. This results in reduced GMP production time and costs, reduced cell dose, a higher percentage of CAR T cells in the dose, increased persistence. And we actually have the capacity to do this in adults, and we know that children are actually a really good target population with this type of therapy because they have enriched naive T cells circulating. I just want to thank everybody who contributed to this project. There's a very large combination of people, but in particular for these two projects, Deborah Mayron, who's listed here, and Joe Zhu were key contributors, Sean McDonald and Daniela Tantalo for the site-off data, Jean and Nico, the clinicians for the second and the first trial, and everybody in the Cancer Immunology Program for the last 25 years, cell therapies, the ONJ, Andrew Scott, and our funding bodies. Thank you very much.
Great, thank you, Paul. Absolutely fascinating presentation, and I might ask Joe to also pop on his camera. We've got time for a few questions, and given that Ricardo is also here and has put in a question, I also wanted to welcome Ricardo Dolcetti to his new position. He's a well-known cancer immunotherapist, but he also is interested in the oncogenic potential of viruses, which is what makes me excited too about his recruitment, having worked on HIV previously and its oncogenic potential. So Ricardo, welcome to Melbourne. I just wrote you a quick note. I'm very proud to say we will be COVID-free by the time you come down here. And maybe you might want to, we're well on track. I was so jealous of Brisbane for so long, but now I'm no longer jealous of anything there except their weather. But maybe you want to ask your question directly to Paul, and welcome.
Yeah, thank you, Sharon, for the nice introduction. So thank you very much to Joe for the kind introduction too, of course. I'm really very excited to be part of the vibrant scientific community of the Peter Mac, the Peter Doherty, the VCCC, the Uni of Melbourne. It's really very, very exciting. I look forward to meet you all and start a collaboration. So I would like to ask to Paul, and of course, thank you for his brilliant presentation, if he had the opportunity to check for the possible increased or enhanced epitope spreading induced by the early CAR T cells.
Yeah, I mean, that's a good question. We haven't got that far yet, but it's a great question, actually. We'll need a model system because currently we need an isogenic tumour that doesn't express Lewis Y to actually look at that. These are xenotransplant models, so we can only look at the human T cells we've transferred.
So we do have a mouse model where so black 6 mice that are tolerized to HER2, to ERB2. So that's an immunocompetent model. Obviously, to get epitope spreading, you've got to have a normal repertoire of lymphocytes. So we would have to probably change the specificity to look at that in principle, I'd say, wouldn't you, Paul?
Yeah, absolutely. I mean, that could definitely be done in the transgenic HER2 mouse system. I mean, that's where Paul and Phil looked for their NI paper.
Thank you. We have another question from Amanda Jones, and the question is, if the early CAR T secrete more cytokines, would you expect more problem in patients with a cytokine storm?
Yes, that's a very, very good question. I think the issue at the moment with solid tumours is the reverse. We don't have any evidence. In fact, we've been measuring it. We don't actually have any evidence of cytokine release syndrome in patients.
Why do you think that's the case?
Sorry?
Why is that the case?
Because at the moment in clinical trials, the conventional CAR T cells are simply not effective against solid tumours. They're either not trafficking to the tumour or they're not persisting there in sufficient numbers. I think, to be honest, the major problem is the tumour microenvironment. Most of the very advanced disease that we're treating, metastatic disease, are cold tumours. So they don't actually have the right chemokine trafficking signals for CAR T cells to penetrate.
Yeah. I think the other way to address that is that the dosing with these cells might be quite different. That's another aspect of that may enable you to control cytokine release syndrome. Of course, with any new product, you do a very, very careful dose escalation to make sure that that isn't going to be a problem. And as always, you would have contingency plans to protect the patient with antibodies, for example, that neutralize problematic cytokines, just in case that were necessary. But we would have to be careful, clearly.
I had a question, Paul, about the synergy with your early CAR and anti-PD-1. Did anti-PD-1 also enhance the conventional CAR T cell activity?
We haven't checked in the model system.
In other models it can, right, Paul? In Paul
Absolutely, yeah. So I think the answer is yes, it can. It's just that the early CAR T cells are more effective. So I think what you'll find is that the the conventional CAR T plus anti-PD-1 will drive more CAR T differentiation. At this point, because the conventional CAR T has such a low memory pool, if you keep doing that, they'll just eventually differentiate to exhaustion.
Yeah.
We have another question from Grant MacArthur, and Grant says, I love this question, of course. Joe, there is growing interest in immunotherapy for viral-induced cancers. What opportunities exist for the centre?
Yeah, that's an exciting prospect. And look, as I mentioned, Grant, and thanks for your question and thanks for your support, by the way. Sharon and I have already had some discussions about exploring this area, and I should mention that. I've also had a separate discussion, which Sharon's well aware of, with Professor Peter Revel. And Peter's a very, very interested world expert in Hep B. And I believe is planning to be integrated more closely with the centre as well in terms of his interest in cancer. And I think Hep B and primary hepatocellular cancer is a fantastic opportunity. We've got a very strong program in many virally driven cancers, but I think there's a lot to do there. And given the fantastic expertise of the Doherty, to me, it makes a lot of sense that we might work on some things like that together. I think the whole area of virally driven cancer is incredibly interesting from the immunological viewpoint. I mean, gosh, you've got these massive, huge, juicy targets, right? And you'd think, well, that'll be easy, but of course it ain't. Because the viruses and our immune systems have learned to coexist over generations, over millennia. So if you look at much of the work of Ian Fraser in papillomavirus, for example, same sort of problem. The immunosuppression, which has been learnt over generations through our coexistence and our coevolution, make raising an immune response to a cancer, even if it's got all these amazing antigens, which look easy, much, much, much more difficult than you'd think. So we're very, very keen to have a go, but it will not be easy.
Yeah, great question. Yeah, EBV, HPV,
HPV, UBV, so many things. But I actually think that talk about poor outcome cancers, and we look at the demographic around Melbourne now, and how frequent it is that we have people with with intractable primary hepatocellular cancer. Of course, these days we're all immunised against Hep B, and we've got good therapeutics now for Hep C. But we have many people who have recently arrived in our country, and amongst those communities, hepatocellular cancer, hepatitis B positivity, big problem. I think it'd be fantastic if we could do something to look at those communities and that problem.
And we're just coming up to the end, but last question from Puya Faridi. I think also if this is for Joe, do you have also a plan for peptide vaccination immunotherapy?
The short answer is yes, and there are many trials of immunotherapy going on around the centre. I have had many discussions with others around the country who are very interested in advancing this approach, probably more than us, in the sense that doing a lot of the basic research, you have to in the end you can't cover everything, but Bruce Robinson in Perth is extremely interested in this. There are groups up in Brisbane. Yes, we are. There are probably industry-based trials that will be coming online, and for those, I'd suggest you speak to the clinical people in the various tumour streams. The short answer is yes, but at the moment it's not a really huge focus of our centre, just simply because of the resources we have.
Okay, well, that brings us to the end of today's Monday Lunch live stream webinar. I wanted to thank Paul and Joe for 2 excellent presentations, and also for having me as your guest chair, and thank the audience for tuning in, and we hope to see you soon, and welcome again to Ricardo, and we'll see you in late November. Thanks everyone. Bye.
Thank you.